All right, good morning, everyone. So today we have a mini symposium that's centered on emerging strategies against anti-cancer uh, approaches. And so what we're going to focus on is the different strategies that people have developed in the recent years that in part are metal-based focus, but you'll also see strategies that go outside of that scope, but that are very relevant to how the field is transforming in terms of making things that are more specific and that are not necessarily chemo drug based. And so we have three different speakers today. One is a graduate student in our institution. Another is a postdoctoral research at the medical science campus. And we have a professor from the University of Massachusetts, although he has not logged in just yet. So I will contact him to see if he has any technical issues. But I'm going to go ahead and get started. So our first speaker today is Louis Delino. He is originally from Haiti. He is currently a graduate student in the chemistry department. He is under the mentorship of Dr. Kai Gribenow and also co-mentorship with myself. And so he's going to focus on research that he has been doing in terms of applying cytochrome C, so there's a protein that you all are familiar with now, in an anti-cancer application. All right, Louis, I'll have you take over.
Okay, thank you, Dr. Tinoco, for this amazing presentation.
I'll let you know when you have 2 minutes remaining, okay?
Yeah, okay. Okay, can you actually see my presentation?
Yes.
Okay. Okay. Okay, again, thank you, everybody. Like Dr. Tinoco say, I've been working on I've been doing some work with on cytochrome C. So under his mentorship and Dr. Kai Gribenow mentorship, and I want to point out first and foremost that in those labs, and part of those labs focuses the development the development of therapeutic proteins for cancer applications. What does that mean? That means that in those lab, protein are being engineered, modified in order to increase their therapeutic benefits. Those type of modification usually are made to increase their stabilities, their stability, their targeting property, and also, among others, their cytotoxicity. So cytochrome C is one of those proteins that we've been working on, basically because of its one of its function, which is activating apoptosis in cells. So these proteins, because of this capacity it has to induce apoptosis when it's delivered in cytosol of cell, it has been intensely studied in our lab. So as all you know, that cancer cells, in order to grow into tumor and also and eventually into cancer, so they have to escape this phenomenon, which is called the apoptosis, in order to proliferate, right? And apoptosis is one. Its role is to regulate this uncontrolled proliferation that is occurring in cancer cell. So the strategy of us in a lab using cytochrome C, or one of my strategy, is to deliver exogenous cytochrome C into those cancer cells that keep proliferating, that escaped apoptosis, in order to induce this phenomena that was being escaped by those cells. So we expect that once this exogenous cytochrome C, when it is delivered to the cytosol of the cells, and the apoptosis will be induced, and finally the cell will be dead. So um, like you said, like you probably know that cytochrome C is a small protein of 12 kilodalton that has several functions. One of one of those functions are the apoptosis inducing property that I just talked about. So beside from this, cytochrome C play a role in cellular respiration by by transferring one electron from the complex 3 to complex 4 in order for the cell to to accomplish this phenomenon, which we call cellular respiration. It has been reported also that cytochrome C participate in removal of reactive oxygen species. So this is a very multifunctional protein. But what we are really interested on in our lab is its capacity to induce apoptosis, which we can use for cancer treatment, right? So, but the problem with cytochrome C, like all the other proteins, is not membrane, or most of the other proteins not membrane permeable, and so that means you cannot enter the cell you cannot enter the cell passively. And also, cytochrome C, for cytochrome C, there hasn't been a receptor or receptors that have been reported. So in order to deliver this exogenous cytochrome C in the cell, some type of formulation has to be done, right? Different formulation has been done in the past year, including in our lab and the literature, such as cytochrome C-based nanoparticle, polymer mediated endocytosis of cytochrome C, mesoperic nanoparticle mediated delivery of cytochrome C, and protein such as transferrin mediated endocytosis of cytochrome C. The thing is, for the first one, the problem you have with the first one is most of them, let's say, formulation. If you don't put a targeted ligand in cytochrome C, you most likely will have a delivery of cytochrome C that is not specific. So attaching a ligand chemically, you risk to attach it, especially when you do random chemical attachment of a ligand. You risk to attach this ligand in some part of the protein that might inhibit its apoptosis function. So based on that, so we in the lab we developed hybrid recombinant hybrid formulation, which we basically conjugate, basically synthesize biologically cytochrome C with protoxin in order to deliver it in glioma cells, as you know, as a potential anti-glioma cell drug. So since, like we said, since we talk about cytochrome C, we don't know much about chlorotoxin. Chlorotoxin is a small peptide of 36 amino acid, and that has been primarily isolated in this species of scorpion venom. Um, and what's good about this peptide, it is reported in the literature to specifically bind to specific receptor and you know, that is overexpressed in glioma cells. So several work has reported that this peptide bind wouldn't bind other cells with a greater extent and than a glioma cell than glioma cells. So, but where the debate is where the debate is which type of receptor is that. Several report has been done. Some reports says this is a chloride channel of 72 kilodalton, which specifically overexpressed in glioma cell. And others have been reported that it is matrix metalloproteinase type 2 protein that protoxin is bind to, which is also overexpressed in glioma cell, same as annexin 2. So the debate is on those receptors, but nonetheless, all of them mention that this protein, this peptide, I'm sorry, this peptide receptor is overexpressed in glioma, which would make it a good ligand to target glioma. So based on that, this ligand, this protein, this peptide has been selected as a ligand, which we combine with cytochrome C as the agent that will induce apoptosis or killing the cells. So we can buy both to one to accomplish targeting, and the other will have the function to induce cell killing. So what we did in order to have a homogeneous drug or potential drug, what we did, we take the two genes, the genes of cytochrome C and the gene of cytochrome C and the gene of clotoxin. We combine them and we clone them into a plasmid, which will result in a construct that we will express in bacteria, specifically E. coli. This type of protein production or expression is a cheap way to produce with high yield protein, and with most of the time have the white folding and maintain their function. That is good because you don't want to do any type of chemical modification in protein where you can affect its function. This most likely wouldn't affect its function. So how we do that, we just place the two genes together. We separate the genes with a 6-histidine tag. This is 6-histidine. 6-histidine will be later used to facilitate the purification of the protein, and we put a kinase cleavage site, which is in red right here. The 6-histidine tag in the kinase cleavage site in red right here, and that will allow us later on, if we need to use this part of the hybrid, we can always cleave it and use part of this hybrid. That's a way to purify at least the peptide also. So in order to produce this protein, we have a couple of steps. We have to express this protein in bacteria. We have to express it. protein expression, and then purify it, and further identify whether this protein has been expressed, or identify the protein. So in the first step, protein expression, we just introduced the construct, the plasmid that we have, that we inserted the um, genes of cytochrome C and chlorotoxin. We introduced this protein, this I'm sorry, plasmid into the bacteria, and that means we transform the bacteria with this plasmid. Then when the protein is expressed, we extract the protein, then we purify the protein by chromatography, basically affinity chromatography. We remember the 6 histidine tag that we put, and this histidine tag is supposed to help to this purification process. And
Louis, you
further,
have 3 minutes remaining.
oh, okay. So, and we express the protein, we purify it. So basically, the first figure that you see in the left is the elution profile of the chromatography steps that we have done, and the figures in the middle are to verify the purity after we purify the protein. This is a SDS gel electrophoresis. Um, with this, we verify the purity of the protein, and this figure that you see here is a immunoblotting that tells us that the protein was actually the protein that we purified, because we use a antibody that's specific to this type of hybrid that we use. And like we said, we characterize the protein, and we compare it, the hybrid with uh, with actual recombinant cytochrome C by itself, so that we use as control in all the further characterization. So the mass of cytochrome C by itself usually is 12 kilodalton, which we see in the mass spectroscopy right here, mildly tough. And here is the mildly of the hybrid protein. So which is 17,000 um, Dalton, which in accordance to the mass that we were expecting. So after that, after purification and identification, we do some spectroscopic characterization, where we the protein we saw the structure of the protein is maintained when we compare it. So structure of cytochrome C is maintained when we compare it in with the actual recombinant cytochrome C by itself. This is a UV vis, and which of the hybrid and the recombinant cytochrome C, that tells us that the heme environment of cytochrome C in the hybrid stays intact. So after structural characterization, we verify thermodynamically how stable is that hybrid when we compare the thermodynamic parameters of the hybrid with the cytochrome C, which we use as control. They were very similar, so
Louis, you
not
have
much
1 minute
modification
remaining.
has been done. So to finish is, after structural characterization, we test this hybrid in glioma cells, and another in NIH treated tree cells. So we saw that these hybrid exhibit very potent cytotoxicity compared to cytochrome C, which we expected to not produce any type of cell death, because it shouldn't be able to enter the cells. Then we verify whether this cytotoxicity was related to apoptotic inducing. So we saw that the caspase evaluation was tell us that this um, hybrid protein were able to induce apoptosis through the caspase activation. So colocalization, intracellular colocalization of a label hybrid in the glioma cell tells us that this protein was actually internalized in the cells and and produce produce this killing. So basically, this is it. In summary, this is a very easy way to synthesize uh, formulation of cytochrome C, and the conjugation of the chlorotoxin, the peptide, didn't affect really the structure of the cytochrome C. Cytochrome C was the apoptotic inducing property was maintained even with and even with this conjugation. Then this hybrid protein is shown very favorable therapeutic index, and I think it should be further study should be done in it so can evaluate whether it can be delivered in actual the brain for
Thank you so much, Lewis. I'm going to
thank
have
you.
the students in the class post their questions, and then I'll have Lewis respond to them in a later time. All right, so we're going to
Okay.
move on to our next speaker. So our
Okay.
next speaker is Dr. Rohit Sharma. Dr. Rohit Sharma graduated from the University of Puerto Rico, Rio Piedras, from the Environmental Science program. He is currently a postdoctoral research at the Comprehensive Cancer Center, which is in the Medical Science campus, working under the mentorship of Dr. Pablo Vivas. All right, Rohit.
Thank you, Dr. Tinogo, for the introduction, and good evening from India, and good morning to all of you there. And let me just share my screen. Uh, can you see the screen, please?
Yes, looks good.
Okay, let me turn on the pointer as well. Okay, all set. So, I'm working in the Comprehensive Cancer Center, as Dr. Tinoco just mentioned, in the laboratory of Professor Pablo Vivas. The lab is specialized in the molecular biology techniques, and especially with the RNAs and the microRNAs for the ovarian cancer point of view. So recently, I have started working on the project, which is CRISPR-Cas9 mediated RBPMS knockout promotes the cell proliferation and reduces cisplatin sensitivity in ovarian cancer cell. So in this headline, you can see I marked three things in the red: the CRISPR-Cas9, RBPMS, and ovarian cancer. So this is the basic the theme of my talk. Like, we have the problem here, which is the ovarian cancer, and the cause of this problem is RBPMS protein, and we are trying to uh, challenge this problem using the tool of the CRISPR-Cas9. So I will go very briefly and explain you that what I'm doing here in this lab. So let's just go for the very basic. What's the ovarian cancer? So ovarian cancer is when the abnormal cells, like all other type of cancer also, which is true in all other cancer as well, in the ovary begins to grow and divides uncontrolled way and eventually form the growth of the tumor. As you can see in this figure, these are the normal cells and these are the cancer cells which start growing. So out of the all the cancers uh, that we have in the world, the ovarian cancer is comes under the top 10 most common cancers in the United States as well as in the world, and is the fifth leading cause of death of the cancer among the individuals. So if you go by the race, 11.5% white people and 10.3% Hispanic peoples, they are the more prone for the ovarian cancer because of the certain genes that are expressed in this population. So ovarian cancer remains like the significant cause of the cancer death, approximately 22,000 new cases and around 14,000 deaths predicted last year in the US alone. So, out of the various ovarian cancers that we have recognized, epithelial ovarian carcinoma is the most common type of the cancers we are dealing with. And out of epithelial ovarian carcinoma, high-grade serous ovarian cancer is the most represent cancer, which is like 70%, and it's very challenging cancer to treat with the traditional drugs and the therapies that we have. So the question arises here: we have cancer and we have therapies, then why we are going for the CRISPR, and why don't we go for the traditional treatments like radiation therapy or chemotherapy? So the if you see here, like once you start giving the dose of the chemotherapy or other drugs, which is like cisplatin, uh, the patient starts having resistance to these therapies. So that means after a certain dose, the patients do not respond to the treatment. And we need to find out what are the molecular pathways which are involved for this resistance in the ovarian cancer. So that's the point where the CRISPR comes and the RBPMS proteins comes into the figure. So if you see here in this very easy figure, I try to explain you that we have the ovarian cancer here in the left panel, and these are the cancers which are the platinum resistance cancer. So what we found here in this cancer, that the RBPMS protein is down-regulated in this two type of cancer. And how we found that? We found on the previous experiments and the previous findings that our lab published. So you can see in this figure, this is the normal ovary here, and this is the tumor. This is the cancerous ovarian cancer. So the difference here in this, that the RBPMS expression is quite high in the normal ovary compared to the cancer ovary. The same finding can be seen here in this figure by using the immunohistostaining. You can see we stain the RBPMS protein, and it was nicely stained in the normal ovary as compared to the B figure, which is the ovarian cancer samples taken from the two different patients. And the same findings were shown in this bar graph, and you can see that the intensity in the normal ovary, the expression is very high of RBPMS as compared to uh, the tumor patient. So, having said that, uh, we find out that RNA binding protein of multiple splicing, which is the full form of the RBPMS, is responsible for the causing the tumor in the patient. So what is the RBPMS actually is? So if you see that RBPMS is a very important protein which controls the gene expression at virtually every step, and it's indirectly linked with the transcription factor, which I'm showing here. So RBPMS is bind to the nascent RNA, and it controls the expression of the genes virtually every step during the process. So apart from controlling the expression of gene, it also involves in the transcription factor proteins, which are CFOS, SMD3, and the CJUN. So these are very complex kind of machinery system of the transcription factor proteins, and any upregulation or downregulation of RBPMS leads to the differentiation, proliferation, and apoptosis, which cause the dysregulation of the transcription factor protein, and it can cause the cancer. So all these findings fall with the previous finding that our lab find, that RBPMS is downregulated in the tumor of the ovarian cancer. So what it tells you? It tells you that RBPMS is a tumor suppression protein. It suppressed the protein at somehow. So that was the hypothesis of our project, and that is how we designed uh, this whole project. We deleted the RBPMS. We knock out the RBPMS proteins from the ovarian cancer, and we found that the cell proliferation was increased, and it reduced the sensitivity of the cisplatin. So how we perform this experiment? So let me give you some very brief overview of the CRISPR and the Cas9. So as you know that CRISPR was studied very recently, and in 2012, and the last year, the Jennifer Doudna got the Nobel Prize and the colleague in the CRISPR field. So it was the discovery that CRISPR originates from the bacteria, and it's kind of the immune systems that bacteria have. So what does it mean? It means that when the virus attack to the bacteria, bacteria take the part of the DNA or the genome of the virus and keep the copy of the genome in their system. So when there is another attack on the virus, uh, they have the copy of their previous attack system, and they develop the system of CRISPR and delete the whole uh, uh, DNA or RNA of the virus, and that is how the virus survive. So you can try to understand this in the very small easy figure. So this is the first step. When the virus attack to the bacteria, this is the attack here. They take the copy in their genome and the plasmid, and this they use this as an antibody, you can say. And when there is a second attack of the same virus, they already have the information in their machinery, and they can design the guide RNA which can cleave the DNA of the virus in the second time of attack, and that is how the bacteria survive. So the Jennifer Doudna and the colleagues and many other scientists, they found out this mechanism, and they thought that this is very competitive mechanism and can be used for the treatment of the various diseases, and that is how they exploit the CRISPR for using the cancer and various other fields. So just want to give you like how the CRISPR works. So there are the two very important components for the CRISPR. The one is the Cas9 enzyme itself, and another is the guide RNA. So you can try to understand this like a GPS system. If you want to go at some place, you need some kind of navigation. So that is how the guide RNA works. The guide RNA guide the Cas9 enzyme where it need to go and cleave the site of the DNA. So this is the guide RNA, and it binds to the target sequence of the in the genome, and the Cas9 come, and then it cleave the DNA to the specific sequence. Now there are some important components which are involved in all this process, and one of the most important component of this system is this PAM sequence. You can see here. You can understand this is like the genomic DNA. This is the single guide RNA molecule, and the blue circle you can see this is the Cas9 protein itself. So what happens, like, you want to cleave the DNA from this particular location. So this so the guide RNA will search. It's like the screen the whole genome and will try to find this PAM sequence into the genome. When it finds the complementary match with this PAM sequence, it will sit here. The Cas9 protein will come and will cleave the DNA, making a double strand break, and the particular gene will be deleted during this process. So as you know, when the DNA is cleaved, the cell try to repair the DNA itself. And when the cell try to repair the DNA, there might be the chances of the mutation in the genome. It might be the insertions or the deletion, which are called indel in the short. And the indel can give the problems of mutations, and that's why the certain problems arises. But the good thing about this, once you cleave the DNA, and the particular sequence was cleaved from this site, you have now opportunity to introduce the new sequence into the DNA, and that is how you can modify the genome, and you can add the DNA from the outside, and you can try to cure the certain diseases which need the gene silencing, or need the gene repair system. So this is how the CRISPR-Cas9 system works very briefly, and that is how we adopted this system for knockout of the RBBMS protein, which I told you it's responsible for causing the ovarian cancer. So we use a software. The software was breaking Cas software, and the software gives the match of the guide RNAs in the left side you can see we got the various guide RNAs, and we selected the best two guide RNAs based on their off-target score. What does it mean? That means these are the these are the best guide RNAs which will do the off-target effect in the genome. And we found this based on the score we get from the software. And the Cas9 protein and this guide RNAs were all put in together in the PSB Cas9 3 plasmid, which is PX459, available very frequently, and it's very efficient system for the transfection of the cell.
Okay, Rohit, you
So
have 3 minutes
yes,
remaining.
okay. So this is the experiment that I'm showing here, that RBPMS protein expression in the clones. So after the transfection of the cells, we deleted the gene of interest, RBPMS. As you can see here, we got the various clone. This is clone 1, 2, 3, 4, 5, 6, 7, and this is the wild type. So in the wild type, you can see the protein is largely intact, but after the knockout, the protein expression was down regulated, and we selected the best clone based on the RBPMS samples and the graph on the left side, which is showing the intensity of the each clones using the densitometric analysis. The same experiment was performed using the mRNA, and all mRNA was run on the gel, and you can see this particular clone was the best one. As you can see, the expression was down regulated completely, and these are the two controls which have the protein RBPMS intact here. The another clone, the expression was there, but it was less than the wild type. So these two clones were selected for the further experiment. After doing the CRISPR, we wanted to know whether we have the mutation is monoallelic or biallelic, and for that we performed some experiment, and we did the Cas9 cleavage assay experiment. For that, we already the kits are available, and you just need to perform the experiment for that, and you need to design the primers based on the region which you are trying to amplifying here. So this is the region. I will go directly to the real experiment. So what I'm showing you here, this is the exons. This is the target sequence. The red color is the exon which we targeted for the CRISPR experiment, and these are the forward primer and the reverse primer here. And the dark red colors are showing the guide RNAs. This RNAs which we selected here. Again, we run the gel, and we found that after doing the Cas9 cleavage assay, we got the two bands, and we found there was a monoallelic deletions in the cells, and the CRISPR experiment was successfully executed, which was confirmed by using the densitometric analysis. We checked the intensity of the band and compared with the controls, and we found that we successfully deleted the protein. So after doing these experiments, once we confirm that the CRISPR experiment was successfully executed, we perform some biological experiment. And you can see here, this is the colony formation assay. In the colony formation assay, as compared to the control cell, the number of cell number was increased after the post CRISPR experiment. So this confirmed our hypothesis that RBPMS is a tumor suppressor gene. So because we delete this gene from the protein, the tumor becomes more invasive, and that's why the number of colonies were increased. The same result was found in the invasion. And we also perform another experiment, which is the senescence, which tells you the metabolism of the cells. And generally, after doing the CRISPR, the metabolism in the cell increases, which was confirmed by using this experiment, as the beta galactoside level was increased as compared to the control than our CRISPR clones. Because we are doing a lot of changes in the protein network itself, so we wanted to see what's the effect on the protein-protein interaction inside the cell. So we performed the mass uh, LC-MS analysis of whole proteins which were present in all the 4 samples. And I loaded this sequence into the software, which is Ingenuity Pathway Analysis, and we found this very interesting network. I'm not going into the detail of that because it's very complicated, but this is the kind of network we got uh, after performing this kind of experiment, and then we shortlist what important connection you can get, and then based on that you can find out. But I will go to this figure. You can see here, this is the RBPMS protein, and these are the some proteins which are directly linked with our interest of protein. And the green ones are the down-regulated protein, and the red ones are the up-regulated proteins, which we found after the MS experiment. So what it tells you that all these proteins have something to do with the cancer. These are either increases or down-regulated, but most of them have the same tendency of RBPMS protein. And for example, this ADM5 protein is the multi-domain metalloprotein that is upregulated in several solid malignancies, including the prostate and the breast cancer. And its expression is also being correlated with the metastatic progression of this tumor. So it makes sense because that is what we were looking for, that the cancer becomes more invasive once we knock out the RBPMS protein. So all these experiments fall in the line, and we clearly shows that RBPMS is the protein which suppress the tumor in the ovarian cell. So that is all I have been doing very briefly. If you have some questions, most welcome.
All right, Rohit. We're going to hold off questions. So I have had the students are going to send me their questions so I can have you address them. Thank you so much for your research. It's very interesting, especially the application of CRISPR. It's great.
Thank you so much. Thank you so much.
Right. So now we'll move on to our final speaker today. So our final speaker is Dr. Rashid Skuda from the University of Massachusetts Amherst campus. He obtained his PhD from McGill University. He did his postdoctoral research in Canada. Canadian postdoctoral fellowship at Columbia University in the chemistry department. And now he is a professor at the University of Massachusetts. He is a bioorganic chemist. And so just very briefly, the work that he has been doing has been centered on understanding at the molecular level the function of potential drug targets. So his lab really focuses on the design, the synthesis of compounds with specific predicted properties and better drug-like properties via structure-activity relationship. So his research combines computational chemistry, medicinal chemistry, and the SAR studies with protein chemistry and molecular biology. So with that introduction, I would like to turn it over to Professor Skuda.
Hey, thank you very much for the invitation. I am very new to this Google Meet. In fact, I asked my daughter to help me to connect. Here we use Zoom, so I apologize of the delay to get in. So how do we do this? I like to...
Yeah, that's perfect. And I'll let you know, so we roughly have 30 minutes and then a few minutes to then discuss a few things that you wanted to do to the students. I'll let you know when you have a few minutes remaining.
The question, I don't see people or the chat. I see only my slide from my side.
Unfortunately, you will only see your presentation, but I'll let you know if there's any glitches.
Okay. Yeah. Thanks very much for the invitation, and I'm very delighted to be here and try to drag you through some of the exciting research that I am going to tell you. Also, because this is a class with mostly young students, I'd like you to also show you a pathway of a scientist, and that will be very helpful for all of you, especially for students who are interested to do research. So I'll start as follows. These two scientists, one of them, Professor Niccolo, who is organic chemist, and Professor Stuart Schreiber, who is a biologist, together stated this back in 1994, where they decided to state it clearly. It is increasingly clear that chemists and biologists have an enormous amount to say to each other. So let's communicate. And today my talk will be a little bit of chemistry using transition metal from my PhD at McGill University, and then medicinal chemistry where I learned biology and cell culture. And here we are. We would like to communicate chemistry and biology and bridge the dialogue to solve a biological problem. So let me start with this, and I'd love to have some interaction with students. So I don't see you. I could see you, but I'd like you to unmute and answer to some question. This is my way of engaging you today. So try to respond when I ask a question. So the first thing I come up with as an analogy is, how do we make cars in general? And I just did my search quickly, and I find out that cars are actually made from something very common to how a human is. So basically, this table is showing since the 70s to now the day, metals are still up to 50% in a normal car. And a car in general is made of copper. This is one of the transition metal I used, and also iron that I will be talking about. So the second thing I'd like to bring to your attention, that our body, and this is me, by the way, uh, it is highly important to recognize what is your body made of. And I just went to Wikipedia and search. So I also showed you how you can make searching ideas and clarify some information through search available to you too. So beside oxygen, carbon, hydrogen, nitrogen, that are higher level percentage more than one, there are a few elements that the body is need to use: phosphor, potassium, sulfur. And I'd like to attract your attention to the bottom side, where is less than 1% of the body, but mainly there are some metals here: chromium, boron, cobalt, copper, fluorine, and those are very important metals. In fact, a recent review in 2019 showed that some of the more than 20 or similar up to 20 metals are important, are essential for the body. And let me drive you through. I told you earlier that bulk biological element, we have the lipid, proteins, and the other stuff, enzymes. So they contain hydrogen, carbon, oxygen. Those are trace. The second category is trace elements that are believed to be essential: magnesium, iron, cobalt, copper, zinc, and molybdenum, and other possibly trace are essential for the function of the body. So again, why I did show you the car? In order for your car to function, it needs some essential element, including the engine and all that. Why the body needs to function properly? You need some important elements, including what I'm mentioning here. Although they are less than 1% of your body weight, they are essential for the function of the human being. So what's so special about these metals? One, I can tell you, structure properties. Metal centers have the ability to organize through interaction with ligand in a wide range, and I'm showing here, and I'm sure your professor already talked about this in your class: octahedral, tetrahedral, square planing, and these type of coordination chemistry with the metal allow you to have a different structure properties. The second thing is reactivity of metals, depending, and these metals have very versatile reactivity depending on their oxidation state. And I'm just showing here a heme binding to an iron, as you might know, which is important in your hemoglobin, for example. So if I have to ask you and interact with you longer, but we don't have enough time, what are the known FDA-approved metal-based drugs that you might know? Anyone can unmute and tell me? I'd like to engage, like I said earlier. Do you know any FDA-approved drug, old, recently heard about it, involved metals, like I said, copper, zinc, others? If not, I will start. This one here is a most old drug-based metal, cisplatin. And I will not go through all the history, but I love to tell some of the history, how this was discovered. Accidentally was discovered by a scientist called Barnett Rosenberg in 1965. And then the idea here is this drug started to be more famous as it shows some interesting data since the 60s. Overall, what I'd like you to take with you is cisplatin, which is one of the metals or cisplatin compound, are prescribed as of today for more than 10 to 20% of all cancer patients. So it is very important to understand and value the metals in our body and how scientists nowadays are trying to cure or eradicate the bad cells in your body, in our body in general, using transition method. How the mechanism works with cisplatin, I know. Quickly, it intercalates the DNA and through several pathways called the apoptosis, intrinsic apoptosis cell pathway, generate the cell death and kill cancer cells. For the sake of the time and for clarification, I have this video to share with you. Hopefully you can hear the sound. If not, we will move forward. So please let me know if you hear the video.
Yes, we can hear it. So Rashid, the sound seems to have no longer going through.
Okay, I'll stop here. I will share with you the YouTube slide and we will move forward. Thank you very much. I hear it very well on my side. Anyway, it shows in a 3D how cisplatin is acting, how it goes through the lipid, how it intercalates the DNA, how it binds the DNA, and how it kills cells in general, or cancer cells. So moving forward, and most of you is smiling, yes, this is a periodic table, and as a chemist, I love to show it each time. It is what highlighted is in green is your transition metals that you must know, actually, at least the first two rows. What highlighted in red are the transition metals that I personally had to work with. Copper, silver, gold, and I will show you a few slides from my PhD, and then iron, I will show you a few works that we have done. So again, as a chemist, because I cannot deny this, I am a chemist 100%. We made compound. I would like to make some compound mimicking the nature. Nature give us a lot of natural products that have high biological impact, although it gives it in a small amount. So extraction of natural product is one of an interest of scientists. But as a chemist, we want to mimic methodology and make compounds that are known in nature. In this slide, and I hope the class environment understand the chemistry that I will talk about, but I will not go deeply in the mechanism. And stop me or if I go toward scope of your class. But in this situation here, you have a single salicyl aldehyde, an alcohol connected to the benzene ring and an aldehyde with an alkyne, and we developed a methodology. This is work done at McGill University under the guidance of Kao-Jun Lee, an organic chemist, catalysis and green chemist, developing methodology in organic solvent. I find out finally that this silver complex to a tricyclohexyl phosphor chloride catalyzed this reaction to give me this intermediate cyclic compound in a Z isomer. This is a double bond in a Z with no purification. We moved forward and oxidized this secondary alcohol to ketone, and this is one of the known boron compound or scaffold was still used to treat thyroid disease. But why we care about that? We develop it in water, a very moderate heat and times, good yield, and also compared to the past synthesis, it requires several steps to make the same desired product. So we achieve a better way, efficient way, 2 steps to get the desired product in organic solvent, which is an aqueous media. Oh, I love to show mechanism, so I have to show that. When you get this catalyst, we believe that the silver is displacing the CH. This pKa of H is 40 with the alkyne, and you have this complex A, and then the salicyl aldehyde come over closely. The silver is oxophilic, like to collect contact with oxygen, and then of the ketone, and then the ortho-oxygen will do an intramolecular cyclization followed by elimination of sulfur. That's our suggested mechanism. For the second work from catalysis, we used gold. At that time, gold was a hot topic to do some CH activation or similar mimicking chemical reaction insertion to the carbonyl and the CH activation. And we find out that gold cyanide and tributyl phosphine allowed us to form this isoflavone in one single step. And we know that those are naturally occurring and used to treat breast cancer. And again, comparative study, you start from cyclic compound to get to the a mixture of the compound. While our case, we did not obtain any mixture. It was a single step from a non-cyclic to a cyclic. So it was interesting to develop methodology to create natural product. Moving from there, we decided to value the methodology we developed, and I expand that on synthesizing additional natural products are shown here. What I'd like is talk about is the pterocarpin, which is a tetracyclic compound, and others. So we use the methodology to create natural products. So that's it for entire PhD that I summarized in a few minutes. Now I'd like to switch the gear to some work that I started learning biology. And again, the word learning, so you're going to learn through your pathway that you decide to choose. So learning is an important way to approach and learn something, go from outside your zone of comfort. So can anybody tell me, I cannot see the chat, how many cells are in the human body? A, 1 trillion, B, 10 trillion, or C, 100 trillion? I think you must know, of course, between the age and the body, but the average number should be an idea, could help you to understand that your body is full of cells. Anyone? Do you see somebody answer in the chat or unmute and just tell me? And in 5 seconds, we will move forward. I hope I can get somebody engaged. I have prizes, by the way, at the end, if you get.
So someone says 10 trillion.
10 trillion. Okay. You tell me the majority. Are they going with B or C or A, for example? And
they're going with B.
B, 10 trillion. Let's do it. Unfortunately, the right answer is C. In general, an adult human being has 100 trillion cells, all originated from a single cell. And the fertilized egg cells in general. So good trial, but keep in mind that your body is full of cells. But wait a minute, how come you have that huge number of cells? And you're still moving, you're still doing everything. So there is a word that called cell death, and there is, the body is trying to, while you're creating cells, others should die, and that's a cycle of life. So cells are always created and destroyed in the human body. But how do cells die? We know apoptosis, and the previous speakers Louis specify apoptosis cell death. So a second question for you, audience, how many cells are replaced on a daily basis? Is it 4 to 5 billion? Is it 40 to 50 or 400 to 500 billion? And then Art, I'll listen to you. If you got any hits or any answers. But every day you have a number of cells getting away, deleted.
We have a
yeah?
C. C. 2 students said C, 1 student said B. 4 C's now.
All right. I like that. Being engaged in class, you learn few interesting ideas time to time. Okay, we'll go with your majority for C, and we'll see. Unfortunately, again, although I try to influence you, we replace around 40 to 50 billion of these cells every day. Thank you for your participation. That was nice. But how do we do that? Okay. In chemical biology, there are different cell death pathway: autophagy, apoptosis, necrosis, and finally, the recent cell death that I will be focusing on in the next slide called ferroptosis. And this was developed at Columbia University, or the first work that we reported is in 2012. But wait a minute, what is ferroptosis? I just said fer and apoptosis. So there is a cell death, well organized, and fer is iron. So here is a transition metal in biomedicine application. So we have an iron involved. The real definition of ferroptosis is a non-apoptotic, iron-dependent oxidative cell death. I'd like you to memorize that or write it down. Just write ferroptosis. It involves an iron dependence, it has an oxidative cell death, and it's not going through a normal apoptotic cell death, which is involving, a normal apoptotic cell death is involving a Cas basis. This one does not involve the Cas basis. So the mode of action of ferroptosis is shown here. And I will show a few molecules moving forward, but this is your membrane. And you have this, I call it the channel or systemic C, if you want more specifications. And you have a cysteine uptake, and you have a glutamate taken off. So there's exchange to keep up with the formation of a cysteine, which is a reduced cysteine version that form the single GSH, and the protein down field is GPX4. When this process is dysregulated, you have a lipid loss. And we notice that small molecules here, erastine is blocking the cysteine uptake. RSL3 is another molecule that blocks GPX4, and FIN56 that is also depleting GPX4. But again, I said ferroptosis. So ferroptosis, to block ferroptosis, you have to add an iron chelator because it's an iron dependent, and you have to add an antioxidant, and I will talk about this in one of the cases called ferrostatin. So you need two biomarkers to prove that it is ferroptosis. Here's the molecules I showed you. Erastine is blocking the uptake of cysteine I mentioned here. And again, I already mentioned a few details about this. This is your ferrostatin 1, the first inhibitor of ferrostatin, and this is the iron chelator we use deform. Again, because we're chemists, I like to show you the carbon binding to nitrogen and others. Erastine was developed a long time ago, 2003, 2004, RSL3, 2010, and FIN56, which is a recent ferroptosis inducer of this subfamily developed in 2016. So again, I mentioned that the cell death of ferroptosis is an iron-dependent reactive oxygen species. How? I'd like to walk you through steps by step of the phantom chemistry. You have an oxygen in your body, and I showed you that you have 65% of your body is oxygen. When you have an electron around, you're forming a superoxide, and that superoxide will react of proton, acid, anything. When you have a pH of your body changes, you have that, and you form a hydrogen peroxide. So somehow you got that formation in the presence of one electron and two hydrogens, you form hydrogen peroxide or H2O2. What happened to that? That will cause you a lipid peroxidation or oxidative stress. But when you have an iron in the body, free, because iron is chelated in the heme, iron free, it will cause an hydroxyl radical. This is again a source of DNA damage, protein, and lipid damage, and that's how we define the phantom chemistry that is involved in lipid peroxidation. So to block that, the lipid damage, you add an iron chelator binding to iron, a deferoxamine or a CPX, which is one of the second iron chelator that we use. To block the radical species, we add an antioxidant or something to mitigate the electron or block or antioxidant reducing equivalent. And we will talk about, Trolox, that's a known one. Endogenous antioxidant is GSH. So I'd like to see if this one will work, and you tell me again. This is a summary exactly of the mechanism of the iron in the cell that cause ferroptosis or cause lipid peroxidation. You can stop me if it's meaningless if you cannot hear anything. Actually, there is a
yeah, we can't really hear it. But
no. I will let it go because you can see the.
Yeah, that's fine. And the students do understand transference, so they will be able to understand this.
Absolutely. Transfer, and there's here an oxidation state happen with this protein and make the iron 3 plus become an iron 2 plus. Here's some of the details I mentioned on the phantom chemistry happening. Looking at the time too. Now, when you have an over lipid peroxide, the GPX4 cannot handle it with GSH, and the system exceeds. And therefore, and that's the main goal of phantom chemistry that I'd like to aware you about it. When you have a little bit of oxidative stress, oxygen is available, you're creating hydrogen peroxide or any other reactive oxygen species, including reactive nitrogen species. That cause cell death through the oxidative stress umbrella. So again, I did mention this again, but now I want to focus on the ferrostatin area. I mentioned if we can prove the ferroptosis cell death, we add an iron chelator to block the iron 2 that came through this way through the iron or transferrin protein that happened through phantom chemistry. So again, the big question here, and I'll show it through a case study, is does ferroptosis occur in vivo? And this is a big question because people are still working on this way. I'll show you what we are at this time. Case study, we would like to talk about the identification of specific ferroptosis cell death inhibitor. I mentioned the inhibitors are the ferrostatin, so I'll talk about that. So it would be great, like apoptosis has a ZVAD as one of the inhibitors of the pathway, to find out a possible ferroptosis inhibitors. That's the idea how we started. So find a drug like inhibitor of ferroptosis. So we call the expert on in silico screening or computer-based. We gather a certain number of compound, and then we created a custom library. We were able to buy it. We use Erastin. Here is the name that I mentioned earlier, a drug that induces ferroptosis. And the question is, could one of these compounds block the Erastin-inducing cell death? And imagine in neurodegeneration, there's a lot of oxidative stress involved. If you find an inhibitor or blocker, that will be very useful and will have a potential applications. So the bottom line, we find 50 analogs, but I'm just sharing with you one that I called FAIR1 or ferrostatin. And this graph is a very standard graph. On the x-axis is the concentration of the compound, and on the y-axis is cell viability. And we use here HT1080, which is a fibrosarcoma, a human fibrosarcoma cell. So when you have a DMSO, no fair add, meaning you have, when you have 5 micromolar of Erastin, you have exactly this things happen, a cell death with this curve. When you have a fair one added to it, you have a blockage of the cell death. And we were able to assess that at an EC50 of 60 nanomolar. So basically, at this level, we were able to prove that when Erastin, this molecule, is killing cells through 24 hours, you get cell death. When you co-treat these two, you're blocking the cell death. And here is the first inhibitor of ferroptosis that was.
So Rashid, you have 3 minutes remaining.
All right. So this is an application. I like to do the chemistry. I show that this is a lipid peroxide, and we show that the mechanism that this fair one is inhibiting the radicals generated there. And we were able to apply this model in collaboration with scientists that has Huntington disease slices available. I would like to show you that I was at UTEP El Paso, and I remember that I drove to Albuquerque to see where exactly this Breaking and Bad movie was filmed. It was exciting. Here's my group. And then that time at UTEP, we were able to solve this problem, these drugs. We're capable now. We generated ones that have a better, improved plasma stability and microsome stability. That's the goal that I would like you to learn from this experience. And then we validate this in an in vivo model with a nephrolog in Germany. Now moving to UMass for a variety of reasons, as soon as we moved, we got the COVID hit. So we did not do too much work, but at least we're safe. I'm glad that everyone is safe. But we have this project that has this compound, very exciting biology, and we're trying to do some chemistry on this. And I don't have a picture with my group. Can you imagine that? Therefore, I'm sharing with you the link from our website. We've been doing a few works, but we hit like everybody. I want to acknowledge my mentors because I mentor, and this is like an interaction. My mentors mentor me to mentor others. So I work under C.G. Lee, Calhoun Lee in Montreal, McGill University, and I work at Columbia University in New York with Ron Broslow, who developed the SAHA drug, and Brent Stockwell and their ferroptosis. So I am very delighted to be one of their students and work under their guidance. For the last few minutes, so bear with me, I promise I'd like to talk about this equity, diversity, and inclusion in academia. And we would love to celebrate this through an inter-university also. So I want to start this conversation with you, and you can drag this information to others and spread the information. A few things that used to be and still going on is stereotyping, implicit bias, microaggression. And you, any of us in this room may experience one of them at least or more. So there is an article, scientists published in 2020 in ACS Central Science, where they come up with 12 principles trainees, PIs, department, and faculty can use to reduce bias and discrimination. I do not need to read all this, but it is for all of us, you and I and others. This is very important moment that we should take advantage and learn about these 12 principles, and the article is here, and also practice them. So bias and discrimination in STEM. Some advice from the article, because I read it. In order to tackle the challenge of bias and discrimination in STEM, those with privilege, the word privilege is important, need to use their power to support and amplify the voices of those with less. And this is how we should work in the future. It is important, it is incumbent on the group leader, you and I, and the audience for future work to demonstrate the importance of considering the EDI and provide education, especially for trainees. So I'm educating myself, I am sharing with you, and hopefully you can educate yourself and share others. What can you do? That's the take-home message. Education, be educative and educate yourself first. Be open-minded to uncomfortable conversation, and this is very important. Do not get impressed with the title of person in front of you, have a simple conversation to clarify things. Be part of this initiative, EDI, in your institution. And for example, if something happened, I think that any one of you who listen to somebody who is in their stress, to say simply, I'm here to listen. If you ever need to talk, or I'm sorry that happened, is there something I can do to help? These are very important advice to help each other and go through. These are situations for calling out discrimination. One of them. Did you notice that you left Jamal off of that email of the group? So corrections, it is hurtful to be left out of things, even accidentally, so we need to be careful about making sure everyone is included. Jose had a great idea. Let us circle back to it for a minute, because sometimes in some groups, Jose or others stated a fantastic idea, but somebody take a lead or stop that conversation and move to another direction. So you should be meaningful and aware about this and bring back to award the person who initiated that great idea. Emily has been doing the ordering for almost a year, and this is happening in your lab, in my lab, in others. It is time for someone else to step up. And then when you make a mistake, I'm not perfect. I know you are not. So when something happened from you, I am really sorry. Sympathize, clearly state that you are sorry about any situation that happened. I did not realize that my joke or my actions could be interpreted that way. Thank you for bringing this to my attention. I will try my best to do better in the future. And again, you're educating yourself, you're sharing that situation with the students, you earn respect. Again, I'm sorry to go over the time, open discussion. I'm happy to continue this conversation today or another time. Thank you for having me.
Well, thank you so much, and thank you for including the inclusivity component of your talk. So if anyone has a quick question for Professor Rashid, feel free to ask. Otherwise, I did email everyone about supplying at least one question per speaker so that we can have some insight in addition to what was presented today.
Yeah, something I want to suggest, but it's not important. Can we all turn our camera on and take a snapshot celebrating this meeting? That would be fantastic. I don't know how to do it, but if everyone can turn their own camera and just, be part of this moment, that would be fantastic.
Israel.
I appreciate that. Very cool. I appreciate that. All right. I don't know how to take a picture. Arthur, take a picture.
Yeah, I'm taking a print screen.
Share it with us. It's amazing to see people connecting from other places, work or other side, but it's great.
Actually, the first time I've seen many of these faces, to be honest.
Yeah. Which is cool.
All right, so yeah, take it. Big smile, one more picture. All right, well, thank you so much to you, to Louis, and also to Rohit for this diverse set of talks. And I hope the students are able to see how many of the fundamental things that we've discussed reach out to these broad applications. All right, then have a great day, everyone, and take care.
Take care. Bye-bye.
Thank you, Dr. Novo. Thank you.
You're welcome.